This link is on slide 53, so make sure that you write down slide 53 and go back and click on this link in the slideshow so that you can watch the animation on protein structure, on the 4 levels of protein structure. All right, so shapes of proteins fall into one of 2 categories. Proteins can be fibrous. In other words, they're just like fibers. They're like strands or fibers. And fibrous proteins are structural, like collagen is a fibrous protein. They are not soluble in water. Most have a tertiary or quaternary structure. And they provide mechanical support and tensile strength. So examples would be keratin, which you find in the outer layers of your epidermis of your skin, elastin, collagen, which is the single most abundant protein in the body, collagen, and then certain contractile fibers like actin and myosin. Then we have globular proteins, which are described as functional proteins, as they typically have, play a functional role rather than a structural one. So examples would be antibodies, also known as immunoglobulins, and they help fight disease, fight pathogens in your body. Protein-based hormones, molecular chaperones, and enzymes tend to be globular proteins. And I would say for the example of molecular chaperones, just so you know what that's referring to, so hemoglobin would be an example because it transports or chaperones oxygen. All right, so sometimes certain conditions in the body, when they're not maintained under homeostasis, denaturation can occur, and denaturation is when the globular proteins unfold and lose their functional 3-dimensional shape, because their shape truly does determine their function. Their 3-dimensional shape determines, and it has to be, specifically that shape, a specific shape, and if it's not the exact shape or conformation that it's supposed to be, then we say it's denatured and it won't function anymore. It won't do what it's supposed to do. Fibrous proteins are more stable. They're not affected by certain conditions that functional proteins are affected by. But for example, the active sites on enzymes become deactivated because their function depends on their shape, and when they're denatured, their shape changes. So what can cause this is decreased pH, which means increased acidity, or increased temperature. So pH and temperature are 2 variables that can definitely affect protein function. Usually, proteins denaturation is reversible if the normal conditions are restored, but if the changes are extreme, then they are irreversible. So although this is kind of hard to give a good example for in the human body, but cooking an egg, you can't undo that. And you wouldn't be able to undo the damage from, for example, proteins that are damaged when a person is burned. That would be an example in the human body. Enzymes are globular proteins that act as biological catalysts, and catalysts increase the rate or the speed of chemical reactions. They do this by lowering the activation energy of the reaction. In other words, the energy needed to start the reaction. And this, because the energy amount of energy is lowered, it causes the reaction to occur at a faster speed or rate. And it allows for millions of reactions per minute. And those enzymes, once they catalyze a reaction, they can catalyze another one. So they're not used up in the process of catalyzing a reaction. Most functional enzymes are referred to as holoenzymes. They consist of 2 parts, an apoenzyme, which is the protein portion, and then a cofactor or a coenzyme. And a cofactor is typically a metal ion like zinc or copper. A coenzyme is, or calcium is a good one. A coenzyme is an organic molecule like a vitamin. So metals tend to be cofactors. Metals, minerals tend to be cofactors. Vitamins tend to be coenzymes. Enzymes are specific to the reaction that they will catalyze. So they act on a very specific substrate or set of reactants. The names of enzymes usually end in ASE, ACE, and they're often named for the reaction they catalyze. So hydrolases, oxidases, there's an enzyme that breaks down sucrose. It's called sucrase. Enzymes that break down proteins are called proteases. Those are easier. Enzyme action is that enzymes lower the activation energy, which is the energy needed to start or initiate a chemical reaction. So here's a graph of the energy required. Here's the energy level in the reactants before they undergo the reaction. And then the energy that's required, the activation energy. Once that energy level is reached, then the reaction will proceed and the products will form, or the product will form. This is without the enzyme, so it's going to take more time because it takes more activation energy. But with the enzyme, that activation energy is lowered, that energy requirement is less, and so it speeds up the reaction. It speeds up the time that it takes for that reaction to occur. This is a picture of a diagram of the way the enzyme works with its substrate. You have 2 amino acids here are the substrates. This enzyme has an active site for both of them. The first step is that the substrates bind at the active site on the enzyme. They temporarily form an enzyme substrate complex. And then in step 2, the enzyme substrate complex undergoes internal rearrangements that form the product. Here, a chemical bond is formed between the 2 amino acids and water is removed. Remember, an H from one of the amino acids and an OH from the other one. So that happens, and then in step 3, the product of the reaction is released. In this case, it's a dipeptide. And the enzyme can catalyze another reaction with those same substrates. It can't catalyze a reaction with different substrates because it is specific to what it will do. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They are the largest molecules in the body. The polymer is the nucleic acid, and the monomers or building blocks are called nucleotides. A nucleotide is composed of a nitrogen base, a pentose sugar, and a phosphate group. I have no idea why every time phosphate is in these slides, you have a capital H here. I don't know why that happens, but I just know that I didn't do it. So I don't want to have to go back and fix everywhere the word phosphate is, but it's not supposed to be that way. Anyway, there are 2 major classes of nucleic acids. There's DNA and RNA. And of course, the word for DNA, the long name for DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. DNA contains the code for our genes, that code for our traits. So the deoxyribo is telling you the sugar. The pentose sugar in DNA is deoxyribose. And then the base can be one of 4 bases. There are 4 bases you can have. There's adenine, thymine, guanine, and cytosine. And then, of course, the third component of each nucleotide is a phosphate group. All right, so DNA holds the genetic blueprint for the synthesis of all proteins. And in that way, since strands of DNA code for specific proteins, in that way, DNA determines our traits or our characteristics. DNA is a double-stranded helical molecule. The shape is called a double helix. It's often referred to as a twisted ladder. DNA is located in the cell nucleus, And the nucleotides of DNA contain deoxyribose, a phosphate group, and then one of 4 bases. They can either be adenine or guanine, which are purines, and they're called purines because they have 2 rings. There are 2 double ring structures. Or the base can be cytosine and thymine. And we can, from this point on, can just call them A, G, C, and T. But C and T are pyrimidines, and they're pyrimidines because they are single ring structures. So, complementary base pairing, it just tells us which base is going to pair with which base. Adenine always pairs with thymine. A always pairs with T. G always pairs with C. So if we know the code in one of the strands of DNA, then we know the code in the other strand because it will follow the complementary base pairing rules. If there's an A, then the other strand will have a T. If there's a T, the other strand will have an A. If there's a C, the other strand will have a G, and so on. And this is the structure of DNA. Let's look at the nucleotide structure first. So in picture A, we have a nucleotide composed of a phosphate, a deoxyribose sugar, and then the base adenine, which is a double ring structure, so it's a purine. The complementary base pairing says that adenine pairs with thymine. Thymine is a pyrimidine. It only has a single ring. So we have 2 nucleotides up here showing how they pair together to form the double-stranded DNA molecule. And then all of the nucleotides in both strands are twisted, and that forms a twisted ladder shape. So the rungs of the ladder, or the steps of the ladder, are composed of the bases, and the sides of the ladder are composed of the sugar and the phosphate. Now, RNA is a single-stranded nucleic acid. And RNA functions in synthesizing proteins. It takes care of some of the steps of protein synthesis. DNA contains the original code, but that code is transferred to RNA, and RNA leaves the nucleus, finds a ribosome, attaches to a ribosome, and produces a protein. So RNA links DNA to protein synthesis. It is single-stranded, mostly active outside the nucleus, contains ribose sugar instead of deoxyribose. Instead of having the base thymine, RNA has the other 3 bases. RNA has A, C, and G, but also U. So the uracil, or the U, replaces thymine. And there are 3 types. There's messenger RNA, which is produced in the nucleus, but then it leaves the nucleus, and it contains the code for a protein. Then there's transfer RNA, which also helps in the process of translation, which is when the code from the messenger RNA is translated into amino acids. And then ribosomal RNA, rRNA, makes up the ribosomes, that forms the ribosomes, which is the structure in the cell where protein synthesis takes place. Now, this is an animation you need to watch. It compares DNA and RNA, compares and contrasts DNA and RNA, and it is on slide 67. And then our last section is on ATP. ATP is the molecule that we produce in our cells, and it is used for energy. The name of ATP is adenosine triphosphate, so that means that it has 3 phosphate groups attached to adenosine. ATP directly powers chemical reactions in cells. And it is produced during cellular respiration when glucose is broken down. Glucose is broken down, and that energy in those chemical bonds is used to produce ATP. And here you see a picture of the structure of ATP. Here's your adenine and your ribose. They form adenosine. And then your 3 phosphate groups. When a phosphate bond is broken and one of these phosphate groups is removed, then that's when the energy is released. And ATP breaks down into ADP plus phosphate by hydrolysis.